Magdalena. Magdalena is a PhD student and Tanya Burkett's lab at Basel University in Switzerland, and she's studying auditory offset responses. Thank you so much for joining today. She's talking today about amplified representation of sound termination in the auditory cortex and its behavior relevance. Thank you again, and we are ready to have your talk. Thanks.
Okay, so thank you very much for this nice introduction and also for hosting this session. So as it was mentioned already before, I will be talking today about auditory offset responses. So when we hear the sound, most of the neurons within our auditory pathway would robustly encode that the sound started, but only a small proportion of cells actually encode that the sound has switched off. So with the first question, we were wondering if there is any clustering of cells showing these offset responses within auditory cortex. So what we did, we compared auditory evoked activity within A1 and AEF cells, and these are two primary auditory regions, and we found that more than 80% of cells within AEF encoded not only nicely that the sound started, but also that the sound was switched off. And having such a region which is so highly involved in sound offset processing, we decided to ask two following questions. So the first one, what are the mechanisms driving these cortical auditory offset responses? And the second, if they are even behaviorally relevant. So to answer these questions, we combined electrophysiology recordings within AEF and its preceding nucleus MGB, together with antidromic stimulation and behavior experiments. So during the first experiment, we recorded neural activity within AEF and MGB evoked by pure tones of 50 milliseconds, and we characterized the temporal dynamics of cell responsive within those two regions. And what we found was that within AEF, more than 80% of cells were actually onset-offset responsive, and when we looked at MGB, onset-offset cells were represented to the same extent as onset-only cells. And this convinced us that AEF is really highly specialized in this sound offset processing. And we are wondering how is it possible that it has bigger ratio of onset-offset responsive cells than its preceding nucleus MGB. So one of the possible hypotheses could be that only these onset-offset responsive cells from MGB are projecting to AEF. And this we decided to check within the next experiment. So to test this, we used antidromic stimulation, where by placing this electric stimulation tip within AEF and then recording electrode within MGB, we were able to evoke antidromic spikes. And then by counting the number, jitter, and the latency, we were able to reveal monosynaptic connections between MGB and AEF. And we found 4 different clusters of cells projecting from MGB to AEF, and the 2 biggest clusters were actually representing onset-offset responsive cells. So in general, we found that more than 80% of inputs coming from MGB to AEF are coming from these onset-offset responsive cells, which indicated that most of this offset information in AEF is inherited from MGB, and that this offset is not fully de novo generated on this cortical level. In the next experiment, we wanted to understand if offset responses are relevant on the behavior level. And to do this, we introduced sound termination detection task. So we trained animals in our head fixed setup, and mice were presented with sounds of duration between 1 and 2 seconds. And then by licking the tube, which was connected to piezo sensor, they had to indicate that they detected that the sound was switched off. So they had this 1 second reward window. And if they performed the lick, then they would receive a reward. So we used this paradigm and trained animals which expressed channel rhodopsin within PV cells. And then within every second trial, we applied laser above AEF. And thanks to this, we were able to activate all the PV cells within AEF region, which in turn suppressed the activity of all the other cells within AEF. And then we assessed what was the effect of this laser suppression on the behavior level. And we found that while comparing animal performance for trials with and without laser, there was a significantly better performance for the trials when the laser was off, meaning that animals were using these offset responses to perform the task. And this was very clear, like in this example session, but was also very stable on the population level. In the last analysis, we were also wondering if we are able to decode the animal decision, if it would perform correct hit or if it would perform a miss, based on the activity from AAF within single trial. So to test this, we used logistic regression, and we trained model and tested it based on different AAF responses. So we use spontaneous, onset, sustained, offset, and late activity. And our model was the best in classifying if the trial was correct or incorrect based on the late responses. But we were quite happy also to see that if we used offset responses to classify the trials, the classifier worked better than when we used spontaneous responses or onset activity. And this again suggested that there is something more within offset responses that could be behaviorally relevant. And with this, I would like to conclude. And what I would like you to remember from this presentation is that offset responses are present both within MGB and within AAF, but AAF seems to be really highly specialized for offset processing. And for the behavior relevance, we show that by suppressing these AAF offset responses, we can affect animal performance, meaning that offset responses can be behaviorally relevant. And with this, I would like to thank my whole group, both present and past members, and thank you for your attention. And I am happy to take any questions.
Thank you so much, Magdalena. That was great talk. And by the way, her recent paper that I think she talked about is in Nature. Like right now, we have in the chat if you want to check it out. The first question is by Joe. He asked that, great talk, the offset response in either MGB-AF diminish over time as the animal is trained the task?
No, it's actually, I think when looking at acute recordings and comparing it with behavior experiments, it's even more robust.
That is interesting. Have you done any histology study on that to see this connection that you are talking about?
Yes, so I think the most interesting thing is that both A1 and AF are receiving direct input from MGB, but I think it's still not very clear how are these cells situated or what's their anatomical organization within MGB. And I tried to address it by building these 3D models of cells recorded within MGB. And the only thing I could see was that onset responsive cells were more in the middle and surrounded by offset responsive cells, but it was not very clear and obvious shape. So they are for sure kind of grouped together, but yeah, it's not that obvious.
And you said that there are also cell classes and MGB, right, that are connected to the AF, right? Or not just a one-on-one connection here.
Yeah. Actually, I had a question. In the behavioral task, is there, I might have missed this because I was answering questions in the chat, but is there, where do the mice respond? So are you worried about maybe motion, especially in the later parts of the trial, contributing to the neural activity that distinguishes between a hit and a miss?
Yeah, so this is exactly what I showed here. And I think I am sure this all late activity is purely motor and reward driven. And we still see a small difference within the peak of offset response. And this is very fast. It happens within 15, 20 milliseconds. So I think it should not be so kind of influenced yet by motor activity.
But I guess, so are mice they're still free to lick around that sound offset?
Yes. Okay. But if they lick just before, then of course the trial is kind of missed. It's blocked and they would not receive the reward.
Ah, okay.
And I think we sometimes see that when we start playing this long sound, that they are kind of preparing themselves. So it could be also that it's kind of motivation and they really are ready to lick. So I cannot fully, I cannot say that it's for sure not motor activity.
Yeah, thank you.
Thank you. There is another question. Bernard asked that are the NGB response types inherited from the IC inferior circuits or not?
So I think they are, but like I did not study circuit further than MGB.
Is that your plan?
Not for now.
Great. Great. Thank you. Thank you again for all the talks, and we are close to wrap it up if there is not any question. Okay, thank you. It seems that that's all. Thank you so much again. We learned a lot about the auditory system, and there are so much promising results that are coming out soon by you guys, and we are very looking forward to that. Thanks again for participating today and all the attendees also. And I hope that you enjoy the rest of Neuromatch Academy. See you around virtually.
Thank you, Roozbeh.
Thank you.